Hello everyone, my name is Sagar Patel and I'm the developer of Riboz ML application. It is an interactive web application designed to analyze bulk RNA-Seq data. It provides traditional bioinformatics results using Lima and Agile packages and also providing options to use machine learning algorithms to predict genes. It is the first of its kind software which allows users to compare the genes predicted from machine learning algorithms and also genes obtained from differential expression analysis. This is the homepage of Riboz ML application, and you will find the information about this application, key features. If you click over here as a result provided, you will see different information about the results that you will get in different tabs that we will see later, and you will see some benefits that this software provides and also the workflow of this software. If you have any information, if you have any feedback or questions, you can just send an email to us over here, and you can click on information to uh, follow any updates regarding to the software. So on the left side, I have a user manual. So these are the different tabs gives you information about how to use this application, how to information about the parameters that you can select and choose to run your applications. So first one is upload files. You can click on these files to prepare your files to run this software, and it is highly recommend to download this data and prepare your files based on this data. Data processing tabs gives you information about the options that you will see in data processing tab. Likewise for differential expression analysis, gene or representation, as well as the gene predictions using ML options. So if I click on pricing, so if you want to use this application, you have to pay per analysis, which means that once you pay, you will be only able to use this application for certain uh, data analysis. So, but if you want to use this application as a monthly, yearly, or for the custom range of time, you can just contact at ribozml@scripting.com. We will be able to provide you custom codes. So now I have to click on pay to use the app, and it will redirect me to the payment option. I will select my details. I will put my details as an email. I will enter my card details. I will enter my address as well. You have to write your correct address. This is a demo version, so now once I have my all information, I just click on pay, and then it will process my payment. Once the payment is processed, you will be redirected to my application, and it will show you about payment confirmation, and you can just click on close over here. Now I will go to upload file options, and I will upload my read count file. I will upload my sample information file, and then I will upload my optional clinical annotation data file over here. Once it is uploaded, I will go through the data pre-processing options. I will just increase these options to 10 and these options to 10. I will select TMM as a normalization method. Then for differential expression analysis, I will select my reference group as a solid tissue normal. This option you will get based on your sample information files. So just create your files as I discussed in user manual. And then I will select p-values as 0.045 and log to fc value as a 1 and adjustment method as a ph. So gene ontology over representation option. My sample is human, so I will just select human. So I would recommend you to select your sample based on the options provided over here. And my gene ID is ensemble, so that's why I have selected ensemble over here, and you can select based on your gene IDs. And I would like to see all sub-ontologies, so that's why I selected all over here, and the rest of the options you can change based on your requirements. So predict genes using machine learning algorithm. This is an optional, but I would select over here so that you will get an idea that how you will get genes predicted from machine learning algorithms. For the options, I highly recommend you to go to the user manual and select and your options based on your requirements. So I have selected 4 machine learning algorithms of models over here, and you will only allowed to use maximum 4 machine learning algorithms per analysis based on the data visualization uh, uh, met uh, to get a better data visualization method results. So now everything is selected and uploaded. I would just click on start analysis, and once I click on that, I will get a message that your data analysis has started, and we will wait to get our results. Once the analysis is complete, you will see these results. And right now I'm looking at the user data information, and I'm at raw read count data. So this gives you information about your data count, the count file that you have uploaded. You have in my data I have 1600 genes, and I have 300 samples. So genes are here as a row, and samples are here as a column. In the second tab, I have sample and group information file that I have uploaded. The ones I have labeled as patient name, and the group type is primarily solid tumor and solid tissue normal. So the third one is raw read count plot. So this gives you a plot of your raw read count file. So you can customize this plot with many options. So I would like to go with this option. So here I have showed you only first 10 samples over here, but if I want to, and this gives you information that total number of samples you have in your files. I have 300, but here I would just write as a 50, so my plot will change to the 50 samples. And I have 3 different options over here: box plot, column plot, and violin plot. And I have 2 options I would see as a count or log to count. I have selected log to count over here, and this is my box plot. If I click on column, it will gives me column plot. If I select on violin, I will get the violin. If I want to see my count data, then I would just select the count and column, then I will see my count as a plot over here. So you can also change the colors over here. I would these are the different color palettes available. So if I select this, my plot will look like this. So you have different options. Also, if you want to if you don't want to see your sample name, you can just deselect this. If you don't want to see your y-axis, you can just deselect this. And you can also change your y-axis label over here. Previously it was sample name, but I can just sample name, I would write over here, change it to sample and all, then it will just change to sample and all. And on the y-axis I have a gene counts written, but I would just change it at genes count all. You can change it to any text that you want. The plot title, I have raw gene count data plot, but I would just I can change it to any text. I would just put data plot, then the plot will change to data plot. And also the legend title, I can also change this as well by just types, then it will change the types as well. So this is options give you to download your plots in different dimensions. So right now I have selected height in inch, which is 8, and width as a 12 in inch. Then I have plot resolution, and if I want to download as a PNG file, I can just select PNG and click on the download plot. I will get my PNG plot over here, which is in 8 by 12. Then if I click PDF and click on download plot, I will get my results at a PDF file, which is very high resolution. And likewise, I can change my, if I want to change my plot size, then let's see, if I want to change to 12, and width I just want to change, let's say for the demonstration, 7. Then if I click on PNG and then click on download again, then the plot will be changed to the due dimensions, as you can see now. So based on your requirements, you can change your dimensions and select your file as PNG and PDF. So this tabs gives you information about the data in your clinical or nutrition file that you have uploaded. I have this group type, race and age in my clinical data, so that's why I can see this tab over here. Now I would like to go to the QC and data normalization results, and the first one is a filtered read count plot results, where you can see the total number of filtered raw reads in data, which is 1598. Previously we had a 1600 raw read count data, and like as described before, you can also change your options over here, and you can see your filter data plot. The other results is filter by expression plot. Now this plot gives information about the total number of filtered rows that you got after filtering step, which is 1541, and this is the plot that you get, and you can modify based on your requirements. And this tabs gives information about before and after filtering data. So right now we want to see first 10 samples. So left side is a raw count plot and the right side is a filtered count plot. So you can see over here that we have selected 10 in our parameters, so that's why we have filtered out those all raw counts in our this filtered counts. As you can see that we had 1600 genes in our original data, and then after filtration we have 1541 total number of genes to further analyze our data. And these are the options that you can select based on your requirements, and edit and save your plots. And the last one is data normalization plot, and where you can see that we have 1541 number of genes, and as you can see our data is very normalized, and it is a box plot, but you can select the column or violin plot based on your choice. You can change the color as well as discussed earlier, and you can save and you can edit your labels and titles based on your requirements. And now we move to the exploratory data analysis, where we have several results. First one, we'll go with PCA plot. This will show you the PCA plot. This is the MDS plot. This is the WOM plot that you'll get, and this is the mean variance trend plot that you will get for your results. Now we'll move to the results, differential expression results, and the first results that we'll get is a differential expression table, where it says that we got total 184 significantly differentially expressed genes in our data. Out of these 57 are upregulated and 127 are downregulated genes. So this is the table that you will get, and if you scroll down, you will see several options. So if I want to download the top hits, the top 10 genes only, I can just write it 10 over here, and click on the download top hits results. So I will get only top 10 differential expression genes if I want to download all the results, then I will just click on download all results. Then it will download all 187 genes. Oh, sorry, 184. And these are the also options if you want to download up and down regulatory genes, if you want to download only up regulated genes, if you want to download only download if you download if you want to download down regulated genes only, you can click on that and you will get your tables. The second one is a volcano plot, which gives you the volcano plot of your data. And here I have two options to turn the significance at descent p and p values. And here you can see the dotted lines, and if you don't want, you can just deselect this one so you won't see those dotted lines. If you don't want to see genes in your data, you would just you can just deselect this one so you don't see any genes in your plot. And the top hits I have selected 10, but if you want to see 20 genes, then you can just replace with the 20, and then you will see the 20 genes in your plot. Also, you can change the text size in the plot. If you want to increase the size, you can just change 15 to 20, then you will see the changes. And also, if you want to see the different colors of your plot, you can just select the different plots, different options over here. And you can download your plots based on your choice over here. And now we'll move to the heat map. So right now we have a total 184 differential expression genes, but in this plot we are only seeing 10 differential expression genes, and we are only seeing 10 samples in this hit plot. And on the right side I have options to change my title, change the font size if I want to. You can see the plot is changed to different text size. And I can also change the different color of my plot, so you can definitely look out these cool colors. You can also change the plot based on your, you can scale on your data. I have selected row over here, but you can select column, or you can select none. And the border color, which is also, I have provided several options where you can put gray, brown, purple. Based on your color combination, you can select whatever you like. So also I have provided different options over here. So if you just deselect this, your cluster rows will be gone. If you deselect this, your cluster column will be gone. If you deselect this, your legends will be gone. If you don't want to see your column or row names, you won't see anything. And remember that we use the clinical annotation file, so that's why we are seeing this age, race, and group type over here. So if I don't want to see it, I can just deselect it, and this is my heat map. I would just select everything, so then you will get an idea that what you're looking at. But right now, what if I want to see my uh, all samples, right? So you would just select 184 over here, and your sample is 300. You will just select 300, and this gives you information about your whole data. But you would just deselect the column name, row name, cluster rows, and the cluster columns, then you will get, and you can also deselect this if you want. But I would just reduce to 100, and genes to like 50, So then you will have some better idea about your heat map. So based on your choice and requirements, you can play around with these cool options and create your heat maps. So now we are going to move with the machine learning results, and I would go with the performance plot. So we have selected 4 machine learning algorithms over here, and the plot gives you the accuracy and kappa result of this performance plot of those 4 models. You can also select different colors, change the titles, and download the plots based on your requirements. The second one is variable importance plot, where you can see the importance of the particular genes that were predicted from this uh, software. And I can also customize this plot by adding more importance value. Like if I want to see dot 20, then the plot will change to dot 20 genes, and I would just change the bar type to the plot type to bar or lollipop. Based on my requirements, I can change that. I can also change the color of my plot if I want to, and it gives you some very cool plot. So I would just reduce to 10 for beta data visualization, and now you can see your plots. And you can also download your plot based on your requirements, and I would go with ROC plot. And also you can see based on your performance, uh, model performance, you can see over here. And also you can download the confusion matrix by clicking over here. So once you click over there, you will get this machine learning confusion matrix, which gives you information about the prediction made by your model and the methods that you have used. I mean the machine learning methods that you have selected. So now we move into machine learning predicted genes. So this results gives you information about the predicted genes using machine learning algorithms. We have 4 machine learning algorithms that we have selected. The RPART model predicted 9 the genes, and the XGB linear model predicted total 37 uh, genes from our data. GLMnet predicted 57, and random forest selected 650 genes. So you can download, if you want to download all machine learning predicted genes, you can just write based on the values over here, and then you can click on the download ML predict genes. You will download all the genes including the importance value. And also the next option that we will go to is machine learning genes heat map plot, where you can see the heat map of the genes that were predicted by the machine learning algorithms. You can change to, you can, if you have different models, I would just go with our random forest. And if I want to see heat map of GLMnet, then I can select that model, and the result will be uploaded based on my choice. So that's why I have selected this. So but if you select R part, and you can see that you have total 9 predicted genes, you will see this. But don't worry, this is just you just have to change to the values that you have predicted. So if you select 9, then you will see the plot. So now you can see the plots over here, and the options are same as you saw in the DE heat map. So based on that, you can edit and save your plots. So this is also interesting results from this application, which is machine learning genes in DE. So right now I have selected the GXP linear model, which predicted 37 machine learning genes. But now what if I want to see that these predicted genes were already in the differential expression genes predicted by G, Lima and AGR packages. So what this table shows you that the machine learning genes, these two machine learning genes in the top 10 differential expression uh, genes table that you have got. So right now you can see the gene ending with 130.16 and 619.14. So if I go back to the gene results, I can see that 19.14 and 30.16, these two genes are here, which were also predicted by the machine learning algorithms. So that's how you can see these results. Also, you can modify these values. So let's say if I want to get the all machine learning gene predicted genes, which is 37, and right now I have total number of genes in differential expression analysis, which is 184. So I would just change 10 to 184, and then I will see that how many genes, the machine learning genes are in the DE results. So I have 13 genes which are common in machine learning and DE as well. That and now I will go to common genes in machine learning and DE, and I can just select different machine learning models. I would just select GSB linear. So this gives you information about machine learning models, the genes that predicted by machine learning model, which is 37, the total number of genes in DE, which is 184, and the common genes M and D is 13. So this is the table that you can get if you want to see the common genes between the two different methods. And also you can download the top 10 or top 13 based on your requirements. And you can also select the different models and see that what other models have predicted the genes. So here this GLMNet model predicted 57 genes, and you got total 21 common genes between ML and DE, and you can see genes and the values over here. And also you can see the common genes heat map, which is based on the values and on the table that you uh, saw in the previous tab results. So this is how you can also generate the heat map of the common genes that were found in machine learning predicted genes and the genes from the E method. And you can also modify the options based on your requirements, and you can save the results. Now we'll move to the gene ontology overrepresentation analysis. So if you have selected ML options, then you will see these three different options for the gene ontology overrepresentation analysis. If you did not select the machine learning algorithm option, then you will only see ORA for the genes only options. So currently it says that you have 184 differential expression analysis uh, differential express genes in your table. If you select this option, then you have to select the machine learning model that you want to see, that you want to get the uh, gene ontology results. And it says that 37, and you can just, if you want to see all, you can just select 37, but I would just and you can go with one by one as well. You can do the gene ontology for differential expression genes, and then you can get the gene ontology results for machine learning predicted genes only. You can also get the gene ontology results for the common genes found in ML and DL. Based on your choice, you can see it, but for the demonstration purpose, I would just go with 184. So I would just select all genes and see what I'll get. And once I'll select what total number of genes I want to select for gene ontology analysis, I would just enter my gene, or just let's say 120, and I would just click on update results. So my gene ontology analysis will start and I will wait to get the results. Once the analysis is completed, I can see my gene ontology results, and I can download these results by clicking over here, and then I will get this table in .csv file format. And this tabs gives you information about the bar plot. You can change the gene ontology category over here, and you can change the font size as well. And then for the dot plot, as we have selected all, so that's why we're getting the all these three different gene ontology results. Gene concept network plot, this gives you information about gene ontology categories, and I have selected 5 categories, which is here 1, 2, 3, 4, and 5. If I want more gene ontology categories, I can just replace with the 10, so then my plot will be changed to those different gene ontologies. I can also change the option. I can deselect it, then I can get this as a network plot, and if I click it, I will get a circular layout. And if I want to color my edges, then I will select these options, then my edges will be colored, and then I will get this output. So based on your choice, you can just select and deselect and change your gene ontology categories. This is the heat plot where you can see different gene ontologies and your genes. Here I have selected 5 gene ontology categories. If I will change to 10, then I will see the 10 gene ontology categories. Enrichment matplot that I can get over here, and I can also change the gene ontology categories. Upset plot, again I can change the category 10 to 15, and the plot will change to the 15 gene ontology categories. This gives you the KEGG results. Right now I have just 600 genes, so that's why I just only got one KEGG results. That I can also download the table by clicking over here, and if I want to see the KEGG plot of this KEGG ID, then I will just go to this tab. I will select the pathway ID, and then I will just click on generate pathway. And it will generate a KEGG plot for us that we can download as a PNG and .pdf file. So now we have got our KEGG plot. We can just click on download PNG, and we can get our KEGG plot. And likewise for PDF, you can click on PDF options. So now everything is done. We would like to generate a report. I am providing 3 different options: PDF, Word, and HTML. You can choose all of them and save it to a file. So let's say with PDF, I would just click on data analysis report download, and it will give me a .pdf file which gives you information about the parameters that I selected for this analysis. I would just open that, and it gives you the method details that were used in this analysis, the options that you have selected for gene ontologies, and the R packages that were used in your data analysis, the options, the parameters that you selected, and the references. I can also select Word, and I can download the Word file as well. This is the Word file, and if I want to download as HTML, then I can just select the HTML, and the HTML will get the HTML file as well. So this is how you get the different results for your data analysis using the Riboz ML. And if you want to uh, reuse or restart the analysis, you can click on the restart Riboz ML application, and then you can select the 4 different machine learning algorithm options. But for that you have to pay again every time for per analysis based on the per analysis charges. So let's say if I want to select the other 4 machine learning algorithms and I want to see that how it goes, so I would just click on this restart Riboz ML application, and then it will start my application again. So before that, I highly recommend you to download your all results, because once you do that, your result won't be stored. So so right now you can see the application is started from scratch, and you have to pay again to and you have to download. Once you have to pay first to use this application. I hope you like it, this application, and if you have any questions, suggestions, you can reach out to me at ribozml@scripting.com. And thank you for your time.